Hello everyone, welcome back. This is Cindy Chen, your MC for this session. In our last session, we heard from over 20 young scientists who shared their research and inspiring ideas. Now we will continue with presentation from more young scientists in the room. Over the next hour and a half, we are going to have other young scientists delivering flash presentation in this breakout room. We will invite presenters to come out one after one. Each of you will be given 3 minutes. Please kindly end your presentation when time is up. Here you can see there's a timer next to me for your easy reference. Without further ado, let's welcome our first presenter. Now please welcome Michelle Yeung.
Thank you. So good afternoon, everyone. This is Michelle from the Chinese University of Hong Kong. The whole story starts when I was walking around the ward and I heard my patients screaming. And guess what? They're doing our gynecological treatment. Now, patients experience pain and stress during the obstetrics and gynecology treatment, including baby delivery, artificial reproductive techniques, and other therapies. So I want to help them by alleviating the pain and stress during the treatment. And guess what I come in mind is Chinese medicine. So as a registered Chinese medicine practitioner in Hong Kong, I think that acupuncture may help this situation. And what we do at first is a market research. So you can't look at these figures. I want to raise a hand who think that acupuncture can help relieve pain. Please raise your hand. Wow, quite a lot. Okay, the next question, who tried acupuncture before? Not much. Now, this aligns with our finding is that over 90% of our interviewees, they agree that acupuncture can relieve pain. But only 20% of them tried because they think that acupuncture is painful. And this is highly correlated. So on one hand, we need to help in education and promotion for acupuncture. And on the other hand, we have some series of research on how to alleviate pain during our clinical treatment. So in here, I use miscarriage as an example. Now, patients suffering from miscarriage, they're in physical pain and psychological stress and mobility. And it actually correlates with depression. This is unwanted. So we first did a meta-analysis showing how acupuncture can help and identified some key acupoints. And then we went RCT, a comparison with sham acupuncture to acupuncture, and it showed that it actually can reduce around 30% of pain and many times of stress, around 20 times of stress. So we think that this result is encouraging when we incorporated the model in more ONG treatments, such as infertility treatment, the oocyte retrieval, putting back the oocyte, that is embryo transfer, and other procedures. And we also building our AI system in suggesting some acupoints for further clinical usage. So this is Michelle Yeung, and many thanks for your time. Thank you.
Thank you for the presentation, Michelle. For the next presenter, we have Fan Shi Nga Miriam.
Dear Professor, fellow colleagues, ladies and gentlemen, good afternoon. I'm Miriam Fan, and I am a PhD candidate from the Chinese University of Hong Kong, and I am also a former lecturer at the Netherlands School of Nursing, a registered nurse, and above all, I am a mommy of an autistic child. So children with autism, they face unique mental health challenges, especially those in inclusive educations, where conventional interventions, usually they conduct indoors, where mainly focusing on the functional and clinical outcomes. However, growing evidence is suggesting the nature-based approach may help address the gap by enhancing the resilience. So the study that I'm presenting today will be the very first study using this approach in our target population, which is autistic children. So we have developed a nurse-led, nature-based, community-based training program grounded in existing evidence, where based on a systematic review, where we find that saline appear to be the most promising contents based on our subgroup analysis. And the protocol is further informed by two studies, one cross-sectional study and another qualitative study, and it was validated by a panel of experts. And this summer, just now, we conducted an assessor-blinded parallel group, a pilot randomized control trial involving 36 autistic children. Half of them were participating in our 6-day sailing program, and with another half participating into our attention control group crafting activity. And the result is very encouraging, where our pilot study showed high feasibility with excellent recruitment, enrollment, and retention rate, where it was highly accepted as reflected by children and their mommy, daddy, demonstrating high satisfaction during our process evaluation. And from our people's effect side, we also indicated a large significant effect size on resilience, though it is a very small scale feasibility trial. We will need a large scale randomized control trial to confirm this finding. So as we speak right now, we are conducting an extended RCT with 184 autistic children, supported by a private donation as well as the Sun Hongay Scalivec Foundations. And to conclude, with continued research and community collaboration, this nurse-led, nature-based sailing program has the potential to become a sustainable strategy to promote their mental health through resilience among these autistic children. And these children, they may appear to be very different, but they are equally important members in our community, especially to the heart of their mommy and daddy. So thank you very much. My name is Miriam. Thank you.
Thank you for your sharing. For the next presenter, we have Rafaela Ares-Lau. Please welcome, ladies and gentlemen.
Hello, everyone. My name is Iris from the Department of Medicine and Therapeutics at the Chinese University of Hong Kong. So today, I'm very happy to talk about sleep problems and a new way to solve sleeping problems through the modulation of gut microbiota. So basically, in the past decade, many scientists have discovered that the tiny bacteria in our gut is actually strongly associated with how well we sleep. And actually, in people who sleep well, they have a lot of good bacteria in the gut. And for those who have insomnia problem, they have a lot of bad bacteria in the gut. So a healthy gut is important for good sleep. So my team thought, what about we transfer good bacteria from good sleeper to those who suffer from sleep problems? And we use a method called fecal microbiota transplantation, FMT. Basically, we collect stool samples from healthy sleepers, and we try to transplant the gut microbes to those who suffer from sleep problems through endoscopy to share the good bacteria. So now let me show you something. So don't worry, for demonstration, this is just instant coffee. So joking aside, we actually tested this concept in 60 people from the Prince of Wales Hospital. So 30 of them actually received FMT through endoscopy at week 2, 4, 8, and 12, and another half of them didn't receive any treatment. And we tried to follow up to observe their mood and also sleep changes. And actually, the results are really amazing because for the primary outcome, we found that 4 in 10 people actually have their insomnia completely resolved after 12 weeks. And through some validated questionnaires, we also found that people who received FMT have reduced insomnia, increased sleep quality, reduced anxiety, and also reduced daytime sleepiness. And we actually also collected stool samples before and after the FMT treatment. And we found that there's a good bacteria called Germiger formicillus that might be important for good sleep. And interestingly, we also found that those who responded to FMT treatment had a very similar gut microbial profile, similar to their stool donors after FMT treatment. So in conclusion, FMT is a really new concept for sleep treatment, and FMT or a bottle of poo might be a powerful tool to treat the sleep problems in the future. So next time, if you want to fix your sleep, remember that the answer may be inside your gut. Thank you.
Much for sharing your insights with us. For the next presenter, we have Abebe Mercuria Shankuti.
Thank you. Good afternoon. My name is Abebe. I'm going to share you about antibiotic resistance and identification of new drug target. Earlier, the bacteria, which we call it Acinetobacter baumannii, was a friend of human being just before 15 years. Recently, the bacteria evolved and began to develop a resistance. And then the major problem of resistance is the formation of biofilm. They exist in community and then try to infect human beings. Then the infection will cause further severe infection. So as you can see, the green one is actually the live bacteria, even if you treat with 1,000 times of the existing antimicrobial like imipenem or carbapenem class. Then, after phenotypic characterization, we proceeded to identify new molecular target, which we call it small RNA 00203. It's a novel target of biofilm formation and antibiotic resistance. So we did in-depth molecular characterization. From that, we identified one marker, which we call it sRNA 003, as a target for as a molecule responsible for biofilm formation antibiotic. Then disruption of this small RNA will result in downregulation of gene associated with biofilm formation and antibiotic, specifically PGA and gene involved in efflux mechanism and the gene involved in cell membrane, bacterial outer membranes. So then after that, after we identified new drug target, we proceeded to screen for the new antimicrobial molecule, which we call it peptide nucleic acid targeting the certain regions of small RNA 2003. This is 53 nucleotide. Along these regions, we target the coding nucleotide along all lengths of small RNA in 5 sectioning, which you can see 5 PNAs, which you call it peptide nucleic acid. From that region, we observed that cell-penetrating PNA in the region of 9 to 23 inhibit biofilm formation in one of the virulent strains of ACB, as you can see on the diagram. Finally, we concluded that the novel PNA targeting small RNA 2003 will be a potential antimicrobial molecule for the combating carbapenem resistance Acinetobacter baumannii. That's all. Thank you.
Thank you for your presentation. For our next presenter, we have Shunsuke Saito.
Hello, good afternoon. I am Shunsuke Saito from Kyoto University in Japan. In eukaryotic cells, newly synthesized secretory or membrane proteins get into the endoplasmic reticulum, ER, and acquire their correct structures with the help from various enzymes and chaperones. However, due to certain kinds of physiological or pathological situations, proteins misfold and start piling up in the ER. To deal with this ER stress, endoplasmic reticulum stress, cells activate several signaling pathways to restore homeostasis in ER. These include PERK, PARK-mediated translational attenuation, and ATO6-driven upregulation of protein folding in the ER, and IRE1-mediated upregulation of protein degradation, protein folding, and protein secretion. Altogether, they are called as unfolded protein response, or UPR, as Dr. Kazutoshi Mori and Dr. Peter Walter explained. And in my study focused on the link between this ER stress and neurodegeneration, specifically in a non-glycosylated mutant of the ER membrane protein, NG-sapin, and that is known to cause dominant motor neuron disease, sapinopathy. In my previous work, we showed that NG-sapin forms an aggregation, and that traps and inactivate the ER calcium ion form SARC2B, drop the calcium ion content in the ER, and that evoke the ER stress and the cell death. And more recently, we have shown that ATO6 branch of the UPR, activation of ATO6 branch of the UPR is cytoprotective for NG-sapin's toxicity. Here's a detail of how it works. Activation of ATO6 pathway transcriptionally upregulates the ER chaperone, CRT, calreticulin. Calreticulin binds calcium in ER in low affinity and high capacity manner. CRT then binds to BEEP, the master chaperone in the ER, and that is also induced by ATO6 pathway. And interaction with BEEP and client protein is enhanced in high calcium ion condition. And by supplying calcium directly to BIP, CRT enhanced the interaction between BIP and energy saving, and that reduced the aggregation of energy saving. Overall, activation of AT6 pathway quite effectively reduced the ER stress by energy saving in this way. And that could help preventing the onset and progression of cerebropathy or other motor neuron disease. Thank you.
Great to learn about your research finding. Thank you very much. For the next presenter, we have Lam Kai Chung Thomas.
Yeah, sorry, I need a few seconds to look at my slide because I accidentally deleted that. So, okay, yeah, and I also apologize. The title is a bit of a clickbait because I haven't actually answered the question, and also from the title, you might be assuming I'm actually working on mice, but actually I'm working on flies, and there are actually a few reasons. So, flies also do a kind of like a reaching behavior. So in our lab, we observed that like when flies are very close to each other, they actually like they push away the other fly. So just like us, they like to like maintain a like personal space. So like don't get too close to me, something like that. So and interestingly, so the like if you quantify the joint angle, it actually correlates with the position of the other fly. So it is a goal-directed limb movement, just like reaching. So basically, and so that's my setup. So I want to like measure the neural activity when the fly is doing reaching. So so that's how it looks like. So basically, like we open a window like on the back, and then record the neural activity when they are doing reaching. So basically, I just stick a fly to a pin and then just rotate it around. So it turns out they also do this kind of like reaching behavior in this setup. And so now I'm quantifying basically the 3D kinematics. So by deep learning, we can track the legs and then do post-estimation inverse kinematics to actually look at the joint angles. And then we can like try to infer which muscles they're actually using. And then we can try to figure out like how this transformation happens from the descending neurons. So descending neurons is basically like an information bottleneck. So there are around like 120,000 neurons in the brain, but there are only 1,000 descending neurons. So basically, it's kind of like information like bottleneck, and then by understanding like what's going on here, so what kind of information is encoded, so you kind of like factorize the problem into like two smaller problems. So and then finally, so if you talk to a fly neurobiologist, it's very likely they will brag about the existence of a connetum. Actually, there are 6 connetums, I think, but anyway. So I plan to like integrate all these information, so kinematics, behavior, and then neuroactivity, and then combine them with the connetum, which is basically a map of how neurons are connected to each other, and then build a model of this whole circuit. So and yeah, if you want to know about more, see some actual data and also like watch some cool videos, so come to my poster tomorrow. Thank you.
Thank you very much for your sharing. For the next presenter, we have Rafael Lesso.
Hello, good afternoon. My name is Rafael Lesso, and I study the importance of randomness in life. So, like Tommy, I also have a poster. If you want to see some real data, come to my poster tomorrow. Parkinson's disease, multiple system atrophy, as well as dementia with Lewy bodies are 3 different clinical diseases with very different symptoms, but then they share the common pathological hallmark of alpha-synuclein protein aggregates in the brain. Duplication, triplication, as well as mutations in the alpha-synuclein coding gene do cause the rare familial Parkinson's disease, showing you the importance of alpha-synuclein, but over 90% of patients do not have mutations in that gene. So these 90% of patients, they have expressed the regular wild-type alpha-synuclein in relatively regular amounts, and yet one protein can cause all these different diseases. And so why is that? We think it's due to what we call conformational strains, where the same protein monomer can actually misfold into different conformations and then aggregate into different strains, and it is these aggregates structures that cause the different diseases. And so one of the questions I asked was then, well, how do different strains even form from the same protein monomer? You can think that maybe it could be due to different cell types where, different proteins, cofactors could be present, or within the same cell type, you could be just different times, different cellular environments because, there's the influx, efflux of calcium ions, for example. Or my favorite hypothesis is random protein misfolding. You keep everything the same, and if you have different strains at the end, there's really no other reason to suspect other than proteins are inherently random. And so this is the topic of my poster. It's important because here it is random. Like that's the thing. I kept the same environment, caused a whole bunch of different strains with different phenotypes, and then that tells the field that hey, your protocol might be problematic if you're thinking that it's the same strain just because you made it the same way. And so that's the first implication. And the second implication goes more to disease modeling, because now as your inherent strain library of multiple strains, you can see one strain looks like Parkinson's, one strain looks like multiple system atrophy, and so we can use these renewable resources that we make in the lab to model diseases as opposed to trying to get access to brain banks and patient samples all the time. So see you at my poster tomorrow. Thank you.
Thank you very much, and great to hear your sharing. For the next presenter, we have Xianglong Hin David.
Hi, I'm David, and I'm from Max Planck in Dresden, from the Marichal Hook Lab. So for many years, the in vitro system is the main driving force when you do modeling different things. But when you try to go into the biomedical research, it often combined with the animal models, disease models. But many of the times, these the preclinical data is not efficient in translating into the human patient outcome. So we need a better strategy to model different kind of cellular behavior and interactions in the lab. So in a lab, one of the key thing is like we try to, so you can see here is a 2D monolayer classic culture of hepatocytes. And when you try to put the cells in a suspension of hydrogel, they start doing some self-aggregation and in a clump of cells, and we call them organoids. So they better mimic what is in vivo in the microstructure as well. So in 2009, in the Hans Clever's lab in Utrecht, they developed the first intestinal organoids from in adult stem cells without using any like immortalization of the cells. And they can start growing this adult lineage of stem cells long term. And since then, we have so many different kinds of organoids growing from different organs. And for example, the brain organoids, people also start growing them from iPSCs. And from that, we can start asking more complicated questions, like how the brain interact with muscles. You can put the brain organoid with muscle organoids, and then you see how the interactions. Or you put more cell types to mimic some kind of niche, and to study the interactions of cells, just like the lab. A lab published the liver per portal assembloids. Or even you can be more adventurous, try with snakes to produce some venom with the organoid. So our lab recently published how to grow human hepatocytes organoid from adult healthy tissues part. And then this can be long-term cultured and allowed the higher throughput of screening as well. And for my work in the lab, I'm using one of the regeneration model we developed in the lab. In this regeneration model, we show that from the adult cells, they can actually develop a similar, when they're growing into organoids, they adopt similar molecular mechanisms like the transcriptome and epigenome to grow into organoids and as a stem cells-like states. So if you want to know more about regeneration, how we apply organoids and the development, come to my poster downstairs as well. Thank you.
Thank you very much for your presentation. For the next presenter, we have Chris Chung.
Good afternoon, everyone. I'm Chris from University of Oxford, and I'm going to talk about my project, which looks at the influenza A matrix 1 protein, which I'll talk about talk as M1 as this goes on. So this protein basically forms a scaffolding inside of the influenza A viruses and forms oligomers that holds up the virus. We know how different conformation works, but conformations of M1 looked, but we don't know how it converts between each other and whether different triggers in this conformation exchange is important. So what I look at is, as a structural biologist, I try to understand the internal dynamics of the protein and try to understand how just movements of the protein in solution actually affects their functionality and in turn and structure as well. So as opposed to what actually mirroring what Professor Walter said in this morning, proteins are not just crystal structures, they're not just snapshots, and they move a lot in solution as well. So I use NMR spectroscopy to look at these structural movements in solution so I can understand the intricacies of them. So as you can see here, basically there are two main structures separated by whether there's an 310 helix in this position or not, and through point mutation and some NMR analysis, I've basically understood that they each of the structure with and without the 310 helix correspond to either the oligomer or the monomer model of influenza A M1 protein. And the idea is that if I make single point mutations, I can basically shut off this equilibrium and disrupt this formation of the M1 helix. And the idea is that if we want to understand, when we understand how these proteins interact with each other, specifically how we can force it into either the monomer or the oligomeric form, then we can have this as a novel way of discovering new antivirals, specifically important for influenza A because of how vaccines vary in efficacy over year to year, and we currently still don't have a global influenza A vaccine, and antiviral resistance will be a thing in the future, just as how AMR was 20 years ago, not really discovered. So yeah, thank you very much. I also have a poster presentation downstairs, so feel free to ask me any questions if you see me just anywhere or like tomorrow during the poster presentation. Thank you.
Great to learn about your findings. Thank you. For the next presenter, we have Chang Nga-Yong Nicole.
So good afternoon, everyone. I'm Nicole Sang, postdoc fellow from the School of Public Health, University of Hong Kong. So today I will talk about our research on transmission of SARS-CoV-2 virus in household setting. So transmission of respiratory infection, like influenza and COVID-19, could happen in different settings. So for example, in social gathering, family, school, or even workplace. So transmission of respiratory infection is particularly quite common in household setting because family members living together, so they have lots of interaction within the household. So in my study, I'm trying to follow up individuals in Hong Kong. So although mild infection may not have significant clinical implication compared to severe diseases, however, they do carry public health implication because they could see secondary transmission in the communities. So in my research, I try to follow them up in the communities. So we have a prospective cohort study of more than 10,000 individuals in Hong Kong. We recruit individuals age over 5 years old in Hong Kong and follow them up and ask them to conduct rapid antigen testing in Hong Kong. So we follow them up, and so that in that case we can identify which individuals in the household is having the first infection. So we can ascertain the sequential order and identify which is the index case and which is the secondary cases. So in terms of results, we find that actually index case that have previous infection before have a lower risk of transmitting the virus to the family members, whereas higher risk of disease transmission is found for individuals that have been symptomatic and also those that are during the pre-symptomatic stages. So as a conclusion, that is our study provide a fundamental assessment of the transmission in the communities and also show that it's feasible to use a household cohort study to identify the transmission chain in the communities. And so we also, because it's some individuals that they could transmit the virus before being symptomatic. So you highlight that you may need the strategy that is irrespective of symptom to control the virus. So thank you very much.
Thank you for your presentation. For the next presenter, we have Yingji Liu.
Thanks. Good afternoon, everyone. I'm Yingji Liu from the Chinese University of Hong Kong. So actually, instead of introduce my study, I kind of doing an advertisement for our Big Mummy research team, which is focusing on the mother and baby microbiome transmission and their later life health. So starting from 2019, we have derived 8 different studies regarding to microbiome, and the first 2 is related to pure or major microbiome, how the early life microbiome developed and how epigenetically these genes can related to the microbiome seeding in a family setting. And then there are 2 environmental or lifestyle-related studies focused on the food additives and also COVID pandemic. As I said, our study started recruitment at 2019, so our whole study is a post-pandemic era study. So all the infants born to our study is after the COVID. So we do find something difference in the infant's microbiome during the study, but how is the long-term effect to these children? Well, I think we can see and wait. And lastly, we have 4 diseases related studies, namely the inflammatory bowel disease models and also the GDM study, which my colleague have discussed earlier in the previous session. And then also Mummy Mind is focused on the neurodevelopmental diseases, and lastly, the atopic diseases. So all of these studies is kind of focused on the more modern lifestyle, which is higher prevalence in Hong Kong and is more related to the westernized lifestyle in their children. And also another advertisement is that my study is focused on the food additives, and my poster is downstairs, so welcome to visit me tomorrow. Thank you.
Thank you very much for your sharing. For the next presenter, we have Wang Shu-Xing.
Hello everyone, I'm Isaac from the Max Planck Institute in Dresden and also from the University of Oxford. And today I'm going to tell you an organelle that is very important for every one of us because it's present in all the cells in our body. It's called centrosome. So centrosome is a classical membrane-less organelle important for nucleating microtubules and orchestrating the cell divisions. And in the panel 1 here is an immunostaining of a dividing cells. So in blue is the microtubules. Okay. So in blue is the DNA, they're condensing because the cell is entering into mitosis. So in green is the microtubules, and they're attached to the chromosome and about to pull them apart as the cell enter into the mitosis. So where all the green line converge to is where the centrosomes are, because they are the major microtubule organizing center and important for maintaining the stability of the microtubules. So for the very long time, people think that it's a very boring organelle because it's static, and also from EM study, it looks like a rock, basically. But from the live cell imaging, we found that it's actually a very interesting organelle that they're constantly changing and remodeling itself for the needs of the cells. So how do we know that it's a very interesting organelle? We use some techniques called the fluorescent recovery after photobleaching, or FRAP. And it's a technique that allows us to shine a very strong light to the structure to deplete the fluorescent molecules. And that will allow us to ask the question, how would the fluorescent molecule replenish when the structure is bleached? So I have videos there showing exactly that techniques. So at the very first flame, you can see that the structure is actually have a lot of fluorescent intensity. But when you shine the light on it, the lower half of the structure disappear, and they gradually reappear because the molecules in the cytoplasm is constantly entering into that part. So on the right side is the CNN, the solid scaffold, and the right side is the tag, is the TAT liquid-like scaffold. As you can see, even the 2 proteins, they localize the same place, they display a very different dynamics. And that graph will show you exactly like this video show. It's actually just by watching at the blue line and quantifying them over time. And you can see the CNN, the recovery starts from the center and gradually flux outward, and it's independent from the unbleached part. Whereas the tag, there's a flux of the fluorescent molecule from the other half, showing the intrinsic dynamics of the tag molecule. So we found that these two scaffolds is actually formed by just one protein that recruiting two different kinases at the same time, that independently triggering the formation of a very solid rock-like scaffold and a very dynamic liquid-like scaffold. And with that, I'd like to thank our very generous funding body, Croucher Foundation, as well as the hosting institutes. So if you're interested in my talk, and I also have a poster downstairs, feel free to talk to me afterwards. Thank you.
Thank you, Isaac. For the next presenter, we have Lo Q Hey Augustine.
Hi, I'm Augustine. I'm a clinician by background and a PhD candidate at the University of Oxford, studying sort of AI applications in healthcare. So today's talk is mostly on machine learning model uncertainty and how it can be useful in a clinical setting. In medicine in real life, I think when we're not sure about something, we want to be a bit more cautious. The problem with classical machine learning model is that they tend to be overconfident because they're trained to classify things, they're trained to put things into buckets, and they're trained to give an answer even when they're not sure. If I say you're at a low risk of having a heart attack, you probably want to know actually how sure are you that I'm in the lowest bucket? Like, is it 51%, 49%? So what I propose here is we use a Bayesian neural network model and use the uncertainty thresholding that we can get from the neural network to make inferences. And there's a sort of retrospective sort of clinical comparison with how does it compare to clinician prescribing. So we use a data set from the Oxfordshire. It accounts for about 1 million patients in the UK. We use electronic health records, develop models, put in essentially what you would get from a mission. And the point is to predict what drug can we use in someone presenting with sepsis. So sepsis is a very severe infection that requires treatment within an hour with high mortality. The problem is we don't know what drug works best until 2-3 days later. So the best guess that we have with clinicians is around 65%. And we use a Bayesian neural network, which differs in a sense that instead of point estimates being propagated along the neural net, it's distributions, so probability distribution along the weights, and the result you get is also a probability distribution. So as you can see here, the median of the distribution constitutes the estimated probability, and the spread of it is an intrinsic measurement of how certain the model is of the results. And by saying, let's say, what if we reject model recommendations if they cross the threshold, if they seem to be uncertain? What we found in sort of C over there is that we were able to increase the prescription rates of giving something sensitive to 80%, prescribe not as broad spectrum, and sort of increase sort of de-escalation antibiotics, which is what we want to prevent more resistance. And what's interesting as well is that by varying the threshold that we use to constrain the model outputs, we can act as actually sort of a trade-off between how well the model performs. It can go up to 90% sensitive, and another trade-off with like how sort of narrow spectrum, broad spectrum describe. Thank you.
Very much. For our next speaker, we have Lu Zhenwen. Let's welcome, ladies and gentlemen.
Do you know that our body can synthesize 10 to the 23rd molecules of proteins every day? And actually, up to 30% of them are misfolded that need to be degraded. But don't worry, our body has a surveillance system that can monitor this misfolded protein. We call it unfolding protein response. Here I'm showing you one of the most well-known unfolding protein response sensor, IRE1. It's a membrane protein spanning in ER membrane. So when unfolding proteins stress happen, IRE1 can directly recognize the misfolded protein and start the oligomerization process. And this oligomerization process actually correlate with its activation. We can directly observe its oligomerization process by using the light microscopy. So when you introduce ER stress to the cell, you can start seeing the IRE1 start from the basal state to start oligomerize forming this punctate structure. And over time, when ER stress gets resolved, IRE1 will go back to the basal state. And this oligomerization process is highly correlated to its activation. So we want to ask a very simple question. What do these foci look like, and how do they get resolved? The way we use is using this in situ structure biology method, which is pioneered by Professor Wolfgang Baumeister. What we did is that we freeze the cell in the cryo condition to preserve its native structure. Then we use a focused ion beam to cut the cell into thin slices. After that, we use light microscopy and electron microscopy to image the IRE1 foci in high resolution at different time points of ER stress. And what we found is like actually at the early time point of ER stress, IRE1 formed a high-order oligomer, forming this double helix and reshape ER membrane to a thin tube. Whereas at the later time point of ER stress, this IRE1 tube end up in lysosome for degradation. Most importantly, if we add a lysosome inhibitor on top of an ER stressor, end up all cell form more IRE1 foci, and this foci cannot be resolved. And most importantly, all the IRE1 foci remain in this state. And you might want to ask, how about its activity? If we add lysosome inhibitor with the ER stress, IRE1 become constitutively active, whereas ER stress itself or lysosome inhibitor itself showing like attenuation and no activity. So if you want to know more about the molecular mechanism from how IRE1 transition from this state to this state, come to my poster tomorrow.
Thank you very much, and great to learn more about it. For our next speaker, we have Shuzhen Wang.
So good afternoon, everyone. I'm Wang Shuren from the Department of Physics from the Hong Kong Baptist University. So today I wanted to give a brief overview about my research group in HKBU. So my research interest is on organic semiconductor materials. So these are carbon-based conjugated molecular materials, and they can conduct electricity. And they have several unique properties. For example, mechanical flexibility, excellent optoelectronic properties, as well as biocompatibility. So it's actually making them interesting or promising for bioelectronic applications. So our work is focused on developing new device structures to develop novel high-performance, high-frequency organic transistors, as well as electrically driven thermal management devices. And what is more important is that these we can also develop sensor materials or sensor devices for different biomolecules or biosignal amplification. And yeah, and they can be actually used for wearable applications for physiological health monitoring. Okay, so this is actually the end of my talk. Thank you.
Thank you very much for your presentation. For the next presenter, we have Jiaying Zhang.
Good afternoon, everyone. My name is Zheng Jiaying. I'm a postdoc fellow from the Chinese University of Hong Kong and Microbiota Eye Center. So my research interest is mainly focused on gut microbiome and gut disease. So today I would like to share one of my projects that is a more translational project, non-invasive microbiome-based diagnostic of inflammatory bowel disease. So inflammatory bowel disease is a gut disorder, and it contains two subtypes. One is ulcerative colitis, UC, and another is Crohn's disease, CD. So now the diagnostic method for inflammatory bowel disease is mainly colonoscopy, but we know that it is an invasive method and also costly. So we would like to develop a new methods that can maybe be more rapid and non-invasive to diagnose IBD. So we collect stool sample from the IBD patient and healthy control, and we extract the microbial DNA, and then we perform the metagenomic sequencing. Then we use the metagenomic sequencing data for analysis and develop a machine learning model, and we identify several bacterial biomarkers that are strongly associated with inflammatory bowel disease. So we test our diagnostic model in a large scale trans-ethnic cohort and validate that they are robust. And also we test our model in other non-IBD diseases to prove that our model is specific to IBD. And another novelty of our finding is that we develop a new technology called multiplex droplet digital PCR. So it's a rapid and accurate tool that can detect the bacterial markers. So we're now trying to develop this all this into a diagnostic test, and hopefully can be used in the clinic. So we are also conducting a large-scale multi-center study, and we hope to collect the stool sample from all over the world and to validate our tests. And that is all of my sharing. Thank you.
Thank you very much. For our next presenter, I would like to invite everyone to welcome Melissa Ranadin.
Thank you very much. It's a big pleasure to be here. I'm currently wrapping up my postdoc also at the Max Planck Institute of Cell Biology and Genetics in Dresden. And next year I will start my own lab at the Ubrecht Institute in the Netherlands on multicellular development. So I'm actually originally a physicist by training. And in my work, I use the physical principles to understand how biological organization works. And a very interesting example of biological organization is early development. So in early embryos, like for example of zebrafish that you see here after fertilization, the cytoplasm is a mixture of biomolecules, proteins, and organelles. And through self-organization, it becomes a regular collection of cells that allow the embryo to proceed to develop. An interesting thing is that after the first division, the cytoplasm is actually already divided into two compartments before the cell membrane ingresses. So this leads to the question of how can the cytoplasm be organized without the presence of a physical barrier that is the cell membrane. So in my postdoc, I found that large microtubule structures that are called microtubule asters fill the space in the embryo, and they drive like this compartmentalization of the cytoplasm. And a very important feature of this process is that in these asters, microtubules can branch from existing ones. So that's called autocatalytic microtubule growth. And I showed that changes of this level of branching of nucleation can drive like different strategies of organizing the embryos. So high level of autocatalytic microtubule growth of these branches leads to very large asters that can fill the embryo in just one cell cycle. And this is a very efficient mechanism because you can imagine that in just like 10-20 minutes, these asters can fill millimeter-sized embryos. Even though it's efficient, this strategy has a downside that it's unstable, and for embryos to develop, this process needs to be very tightly regulated by the cell cycle duration. Low level of this microtubule nucleation, of this microtubule growth that are observed, for example, in fruit fly embryos, lead to a different strategy of like small asters that by multiple divisions can progressively fill the space of the embryo. I mean, I don't have time to go into the mechanism of these strategies, but something that is interesting is that they lead to different timings of cellularization, which is the transition from cytoplasmic organization to multicellular systems. And in my lab, I will study what are the principles of physics and molecules that allow these organizations to emerge, and how the cellular machinery adapts between these two states. Thank you.
Thank you very much for your sharing. For the next presenter, we have Jason Kang Xin.
Hello, everyone. Since there are already have many young scientists talk about the microbiome, I want to crack the identification here. Microbiome study actually is not only about any single bacteria, or we are different from microbiology. So we focus on the community. We work on all the microorganisms, include the bacteria, fungi, virus, and the metabolites produced by all these microorganisms. So my work actually started from Hong Kong. I received my PhD from CU Hong Kong. And about like 3 years ago, I decided to relocate to Singapore. And this microbiome study on the cancer work actually started almost 10 years ago before I joined the team. So actually the work started from the observation, and we observed actually the colon cancer is associated with the microbiome, but the association is different from the causation. So how do we do that? We did FMT. So the FMT transplanted was did from the colon cancer patient to the germ-free mice. This is the very first time we find after the transplantation, the germ-free mice got the colon cancer. So after that, we think, okay, we really need to do some mechanism study. So what we did is we used the human cohort, and we identify different bacteria in the cancer patient, and with the compare with the control, healthy controls. So after that, we think, okay, the next step, we want to translate with what we found to the clinical using. So we have some different diagnostic key get the pattern, and also we are trying to use it in the immunotherapy, chemotherapy. So there's one paper I published during my PhD training. It's a bacteria called Rosabura, and this bacteria can enhance the immunotherapy. So based on all this data and cohort I collected in Hong Kong, I used it to get my first grant from Singapore, and awarded by the NMRC YRG. So I have my own grant, work on the chemotherapy. Again, it's still this very powerful bacteria. So I don't have any actual data or poster to share with you, but if you want to learn how to, as a foreigner, how to get the grant from another country, you can talk to me. Thank you.
Thank you for your sharing. For the next presenter, we have Chao Zheng.
Hi everyone, my name is Chao Zheng, and today I'm going to share with you about my PhD project in Karolinska Institute. My project is focused on oligodendrocytes, which is a type of glial cell in central nervous system. And these cells form the myelin sheath, which is a lipid-enriched structure that wrap around axons, enable the rapid and efficient electronic signal transmission between neurons. And in multiple sclerosis is an autoimmune disease in the central nervous system. And in this disease, our immune system mistakenly attacks myelin. And it will lead to demyelination, disrupt neural signal transmission, and also result in symptoms such as impaired coordination and movement. So in our study, we use the animal model of multiple sclerosis and combine with single-cell multi-omics and spatial transcriptomics to investigate the changes of oligodendrocytes across different stages of disease. And our study found that in this disease, oligodendrocytes can express immune genes, and even at the late stage of disease, they still maintain the chromatin memory of these genes. And these persistent immune epigenetic states may contribute to the worsening of symptoms during the disease relapse. And we also identified the functional heterogeneity within oligodendrocytes. We found that there is a subgroup of them which has enhanced immune feature, while another subtype has a characteristic which is more related to neural repair. And these repair-oriented subtype may represent a promising target for disease remyelination therapy. And in addition, we also apply in situ sequencing to examine the spatial dynamics of oligodendrocytes and other glial cells across different stages of disease. And we found that the interaction between disease-associated and non-disease-associated glial cells may drive the chronic progression of of multiple sclerosis. So overall, our study aims to improve our understanding about oligodendrocytes in multiple sclerosis and also identify the therapeutic targets for it. Thank you all for listening.
Great to learn about your findings. Thank you. Next one, we have Jasmine Kali.
Hi, everyone. When I designed this, I must have thought that we were presenting on a Times Square billboard, so sorry for that. But yeah, so my name is Jasmine. I'm a second-year PhD student at the University of Cambridge, and the focus of my work is within cardio-obstetrics. So cardio, we're looking at cardiac disease, and obstetrics, we're looking at pregnant women. So my question is essentially, how can we best care for this growing population of women who are entering pregnancy either with a pre-existing cardiac disease that's congenital, a pre-existing cardiac disease that they've acquired during their lifetime, or significant cardiac risk factors, which are getting increasingly common as rates of obesity and diabetes and other such comorbidities are rising. So the work that's displayed here is from a systematic review that I conducted, which ended up getting split up into looking at the antenatal and then postnatal experiences of women with cardiac disease. So within the experience of pregnancy as somebody with a cardiac disease, you don't necessarily experience the same care pathway that somebody without this indication would experience. So you have to go to the doctor a lot more often. You're experiencing increased monitoring, and this can be very stressful. So we developed frameworks to understand how women experience this increased monitoring and are now moving into the second phase of this work, which is where we're looking at the actual experiences of a cohort that we have at the Cambridge University hospitals, because we have a specialized cardiac clinic that services the entire east of England, which sees approximately 80 patients per year who are very high risk with cardiac disease. So we now have some preliminary results for the different ways that these women experience their care and the ways that their interactions with providers are influencing their experience of pregnancy. And then we are also taking this cohort and expanding it outward, and looking at a longitudinal multi-year insight into some of the outcomes. So from this, we've generated a number of hypotheses, including some ideas about potential drops in hemoglobin and how that actually having delusional anemia right before delivery may not truly be correlated with adverse outcomes for this specific population of women. And then we are going to be testing that in a national cohort of approximately 2 million pregnancies, 29,000 of which are pertaining to cardiac disease. So we've done kind of the background work and looked at it at the smaller scale cohort that we have with about 400 pregnancies and are now looking to expand this outwards. So if anybody has similar interests, we'd love to speak to you further, but thank you all for your time.
Thank you for your sharing. For our next presenter, we have Alexandra Alpo.
Hello, I'm Alex. I'm a postdoc at the University of Hong Kong. Sorry for my poster. I thought I had to show my poster here. But I'm working on a project that's using aptamers as targeted delivery systems for glioblastoma. So glioblastoma, the survival rate is still only 15 months, so it's very important we have new therapies for this. The current therapies are surgery, radiotherapy, and chemotherapy, but none of these are doing a good enough job. Or we have immunotherapy, which is a bit more new, but we're still having problems of targeting the tumor itself and also crossing the blood-brain barrier. So because of this, we want to come up with some targeted molecules that will go to the tumor and then target the particular cells that we want to target. So the molecules we're using are aptamers, which are small oligonucleotides that fold into 3D shapes, and they have very high binding affinity and specificity to their target of choice, and they also are small enough to cross the blood-brain barrier. So we've made several aptamers, but I'm just going to talk about our T-cell aptamer today. So our aptamer has very high binding affinity for a T-cell surface protein, and when testing the binding affinity, it has a very high binding affinity in the very low nanomolar range. And then when we do biodistribution studies on our glioblastoma mouse, they go straight to the tumor and they go only into the T cells and no other cells around it are affected. So this is great. So cool, what can we do with this now? So then we've conjugated an mRNA onto our aptamer, and we're using MOX40, which is therapeutic mRNA that we want to overstimulate in our T cells. And in our T cells, this will then allow them to interact more with the macrophages and the tumor cells and hopefully stimulate an immune response and help clear the tumor itself. So then we've made this conjugate, and then we've also injected it into our glioblastoma mouse models by IV injection. And we can see that they go to the T cells, and we can see that OX40 is overstimulated on a 20% increase. So this is great so far. And then we're currently looking at other tumor models and also what other things can we deliver in the same way. So if anyone else would like to talk to me about this stuff, I'm very happy to do that. Thank you.
Great to learn about your sharing. Thank you. For our next presenter, we have Ying Xie.
Thank you for the organizer to give me this opportunity to share my postdoctoral research. I'm Xie Ying, a postdoc working in New York University. So imagine a busy factory with all the assembly line running in full speed. Everything works smoothly, but until a disaster strikes. Machine breaks and failed product pile up. To restore order, repair crews has to quickly come to the damaged site. Otherwise, the factory need to shut down and loses money. In every living organism, the individual cell is like a tiny factory, and they can face the same problem. And normally, they are very crowded with proteins and mRNAs, which are essential building blocks to keep cells alive. But under stress, such as nutrient deprivation or attack by pathogen that causes infection, the crowded environment may turn into dangerous because protein within can easily misfold and become toxic aggregate. And the repair crew in the cell called chaperone need to quickly go to the damaged site to refold protein or clear it. Otherwise, the cell may lose viability in long term. So in my postdoctoral study, I try to ask the question, is there any common mechanism that can serve to prevent overcrowding when a cell undergoes stress? And I use live cell imaging techniques, which allows me to watch the proteins or mRNA move in real time. And I also introduced a bright protein nanoparticle, which allows me to measure macromolecular crowding level within. And then by applying genetic and chemical perturbation, I asked what are the processes are important to control crowding. And here is what I discovered. So upon stress, protein translation quickly turned off, and the mRNA will condense into droplets. They can be found as stress granule or processing body. And this actually allows the whole cytoplasm become more fluidized. And the chaperone protein can be easily moved and reduce protein damage. But if I block these processes, the fluidization processes is blocked. And this actually reduce the cell to enter quiescent state. So this study actually tells us that cell can not only using mRNA as a template for protein synthesis, but it also has a direct role for biophysical regulation. Perhaps failure of such regulation could be one of the reasons that causes diseases such as aging or cancer. And thank you for your listening. Yeah.
Thank you for the presentation. For our next presenter, we have Damien Chua.
Good afternoon, everyone. I'm Damien from Singapore, NTU. And today I'll be sharing a little story of fatty liver and my PhD project. So today, increasingly, fatty liver is being recognized as a global silent epidemic. And in a row of 10 people, it is estimated that 3 of us would have fatty liver. And by 2040, it will be 5 of us. So and paradoxically, we know that fatty liver patients have a syndrome called leaky gut syndrome. And here I was very curious on why this happened. So in our lipid model, a mouse that can have fatty liver, we identified that very early on, before fatty liver develops, the gut has been starting to present disease and leaky gut conditions. And here we disentangled 2 pathways where, firstly, through dietary fats, it directly increases gut permeability issues through ANGPTL4 directly through this pathway. And secondly, through gut dysbiosis. So with a fatty diet, you will have gut microbial problems in your gut, and then this causes changes in your molecular patterns of the gut microbiome. Very quickly, we see that there's an increase in stimulator to silent flagellins, causing activation of ANGPTL4 protein, and subsequently the gut permeability issues. We can discuss the deep biological mechanism after this if you're interested, but a question that I was thinking is, how do I translate this from the bedside to the bench and now back to the bedside again. So firstly, we organized a multi-center Asian cohort to look at how fecal ANGPTL4 is changed in fatty liver patients. And we see that across 3 countries, Thailand, China, and also in India, fatty liver patients do have increased in ANGPTL4 proteins in their gut, approximately through the fecal samples. And secondly, we flirt with the idea of having next generation probiotic design for metabolic diseases. So in this case, when we think about probiotics, can we now look at molecular patterns of the gut microbiome instead of species? So this would be a more species agnostic method into personalized microbiome medicine today. So with that, we have also done several proof of concept studies in mice and also in human studies as well. So with that, thank you so much for listening, and I hope to discuss more with people who are interested in my work. Thank you.
Great to know about your sharing. And our next presenter, we have Dickie Wong.
So hi guys. So I'm Dickie from EPFL in Switzerland. So apparently I'm the only synthetic chemist here. So instead of scaring you guys with a lot of hardcore technical chemistry, so let's forget my slides. So do you guys still remember what you have learned in organic chemistry classes? Do you still know what is azides? Yep, I think you should remember, because like a few years ago, there's a Nobel Prize getting in bioorthogonal chemistry. So imagine if we replace nitrogen from azide to 2 carbon. So originally azide is relatively explosive, but it is still stable enough in biomedium. But if we replace it with 2 carbon, that would be super explosive. We have several accidents in our lab that explodes, and somehow I survived. And interestingly, this kind of thing is highly explosive, cannot be isolated for many years, but somehow in our lab, and also one lab in TU Dortmund in Germany, so we installed a very bulky group that is called N-heterocyclic carbene in this functional group, highly explosive thing. We stabilize it, sort of. I mean, it's still air sensitive, water sensitive, but it's stable enough at room temperature under box condition. So it just behaves like azide, actually. You can still do, like, for example, click reaction, etc. So one of my work recently, that is, we try to re-add this kind of explosive thing with like an iridium complex. So iridium complex is very well known as a kind of catalyst to do a lot of organic reactions. So those iridium complexes are actually yellow in color, and my complex, that diazolifin, is also yellow in color. So when we mix it together, it didn't explode, of course, but it turns to blue in color, which is very unusual, because like normally iridium complex should be yellow in color. And I spent a lot of weeks actually to isolate it, make it clean, and I did some chemical modification, and somehow I isolated this kind of complexes. So it seems like it is quite useless or boring, but somehow because we somehow break the textbook rule that this kind of N-heterocyclic validity, we call it as, shouldn't be like stable at room temperature. So somehow by also removing one chloride, we get this kind of, we call it as a pogo stick complex. It's just like what you played in, maybe probably in your childhood. But this 2 type of complex, by just removing one atom, it changed the reactivity completely. So I don't want to go through a lot of details in organic chemistry, but if you are interested in those chemistry or the explosive incident, just find me, yeah.
Thank you for your insights and perspective. For our next presenter, we have Jiaqi Li.
Hi everyone, my name is Jiaqi Li. And so you guys might have already forgotten of your COVID-19 infection or the bad flu that you had last year, but your body never forgets. So there are a group of specialized immune cells that help to maintain immunological memory. And what for, you might ask? It's to maintain, it's to enable a quicker, stronger, and more effective immune response with every subsequent encounter with the microbe. So This system is built into all of us, but some of us feel much worse during an infection while a friend could go about their day as if nothing has happened. And you might think this is luck, but probably not. So here at Karolinska Institute, we are interested in finding out exactly why this is so. So we work at the intersection between clinical virology and immunology. We employ clinical cohorts with unique clinical interest factors such as the young children, healthy adults, the elderly, individuals who are obese, pregnant, asthmatic, and all living with chronic illnesses. So the goal is really to understand how each of these clinical factors can influence the memory T cells, which is one of the major cell types that provide immunological memory. So to do so, we employ methods such as high-dimensional full-spectrum flow cytometry, single-cell omics, antibody and microbiome analysis, as well as bioinformatics to enable a deep, precise, and high-resolution immunoprofiling of the memory T cell responses. So with this pipeline using COVID-19 as an example, a group has shown that T cells against SARS-CoV-2 are established and functional from as young as 2 years of age, and these T cells remain functional well into adulthood, but they decline with age. So that kind of provides a basis as to why we should continue vaccinating the elderly, and it also provides a basis as to why younger adults tend to have less severe COVID-19 disease. So a better understanding of these immunoprofiles of the memory T cells will allow us to know which are the key players that are involved in the different clinical manifestations of these cohorts. And that would enable us to design either new therapeutic targets to improve clinical prognosis or new biomarkers to improve detection and protection. So the next time you or your friend is recommended for a certain clinical precaution, just know that it's in place for good reasons. Thank you for your time.
Thank you very much. For our next presenter, we have Hermione Mark.
Let's talk about food and climate. We are experiencing increasing interconnectivity and emerging risks due to climate change, and placing immense pressure on food systems. And it is clearer than ever that food system plays a very central and inextricable role in the planetary boundary transgressions. The recent Eat Lancet Commission report reveals that food systems are the single most influential driver, breaching 5 of 6 planetary boundaries, and contribute about 30% of the greenhouse gas emissions. Understanding the links between food, including seeds, agriculture, and climate is crucial to reshaping our food systems towards sustainability. Food systems are complex, encompassing production, aggregation, processing, distribution, consumption, and disposal. Transforming food systems requires a people-centered approach focused on everyone's right to adequate and nutritious food. With less than 5 years to the Sustainable Development Goals, progress remains fragile and unequal, particularly in ending hunger and ensuring food security for all. Addressing food systems transformation in the context of climate change is essential for global efforts. Consider the critical role on the scarred global seed belt in safeguarding crop genetic diversity for food security for all. Innovations in agroecology or regenerative agri-food systems are vital for securing our future food supply. Climate-smart agricultural practices, or may be supported by AI and science, can enhance productivity and resilience. Additionally, seaweed, currently emerging as a promising strategy for climate education and mitigation, which offering a potential to advance food security. These innovations can empower communities and promote sustainable stewardship of our resources. In a world where the resources are scarce, food must never be overlooked. Now it's the time to reflect on pathways for sustainable food and agriculture in the face of climate change, and ensuring that future generations have access to adequate and available food choices, to achieving healthy, safe, and nutritious food systems. Thank you.
Thank you very much for your sharing. So here comes to our last presentation. Everyone, let's welcome Jingjing Liu.
Yeah, hello everyone, I'm Liu Jingjing from the HKBU and I'm the postdoc from the Professor Hetauli's group. Our group focused on the developing of the functional metabolomics and applying the functional metabolomics in investigating the molecular mechanisms of the complex diseases from the perspective of metabolism. And today I'd like to talk about how we use the functional metabolomics to develop the biomarkers and identify the therapeutic target of the pancreatic cancer. First, we need to know that if we want to use metabolites as a biomarker, it should be accurately identification. So we spend a lot of time and money to establish a targeted metabolomics method. Now it includes more than 1,000 reference compounds. It means that we can quantitative and qualitative analysis more than 1,000 metabolites and lipids from different kinds of samples like tissues, cells, and plants. So we can see that we collected the clinical samples from the hospital and characterize their metabolic profiles. And then we identified which the metabolites has significantly changed in the plasmas. So based on these stringes, we have found 5 functional metabolites that can combine as a panel to distinguish the pancreatic cancer patients from the healthy controls. And then we know that not all of the biomarkers can become the therapeutic target, Not all of the biomarkers can become the therapeutic targets. So how we identify the functional metabolites? What is the functional metabolites? It means that we can hinder the tumor growth by modify the expression level of these functional metabolites. So we used 2 stringes. The first, we combined our metabolomics with the proteomics and RNA sequencing analysis to construct a functional network related to the pancreatic cancer. So it helped us to easily pick up the key nodes. And the other stringy is that we use the effective drugs as the fish hook to identify which metabolites had significant change in the disease state, but recovered when we treat with the effective drugs. So based on these 2 stringes, we have identified 2 therapeutic targets. The first is citrate metabolism, and the other is the AMP-CMP exercise. So now we want to update our functional metabolomics into a single cell level. So we warmly welcome everyone here that calculate and connect with us. Thank you, and have a good night.
Thank you very much. So once again, I would like to invite all of you to give the big applause for all the presenters and also the participants who are joining our sessions. We have listened to 27 very innovative and inspiring ideas. I believe that you might have found your interest in some presented subject or in some ways that you were inspired with the new ideas. Our heartfelt thank you to all the presenters for their contributions. For our honorable laureates and forum participants who have enrolled for the dinner, we have arranged coaches to transport everyone to the peak. Please arrive to coach boarding area at building 15W before 5:45 p.m. Should you need any assistance, please approach our event staff. Everyone, thank you and have a nice evening.